Thank you very much indeed for that kind introduction. I feel as though it's a great privilege to be here, to be speaking at this 11th event of the Future of Science program. I hope it goes on for many more years. It's obviously an extraordinarily exciting project that I hope you can keep going with many interesting speakers in the future on a whole variety of topics. And I also want to say what a pleasure it is to be back in the magical city of Venezia. I think this is my third, at least my third visit, and it's always delightful to come back here again. So my talk today is quite a bit different from the previous talks. First of all, I'm a social scientist, and secondly, part of the approach that I take to this big enterprise of precision medicine is one in which, as a social scientist, I am in many ways much more interested in the causes of ill health and disease, the causes of longevity, why some people do so much better than others in terms of lifespan. And I am, although obviously developing therapies, developing treatments is absolutely crucial, and we've heard remarkable talks today about the progress that is being made in precision medicine. In some ways, what I have to say today will be a bit of a counter, not to displace anything that's been said in any way whatsoever, but to point out that we have taken off a very large bite, and it's an extraordinarily complex project, and there is more to it than simply developing therapies, although obviously that is of prime importance. So I'm left-handed, so I have to switch everything over here. I want to go back to Francis Collins and what he had to say in Time magazine in 2003. He, as many of you probably know, is the director of the National Institutes of Health in the United States. And this is what he had to say in 2003 about precision medicine. I think it's safe to say that we'll have individualized preventive medical care based on our own predicted risk of disease as assessed by looking at our DNA. By then, each of us will have had our genome sequenced because it will cost less than $100 to do that. As we've heard, we haven't quite got that far. And this information will be part of our medical record because we'll still get sick, we'll still need drugs, but these will be tailored to our individual needs. They'll be based on a new breed of designer drugs with very high efficacy and very low toxicity, many of them predicted by computer models. Now, just as Francis Collins published this statement, findings from the Human Genome Project signaled a red alert to some scientists. In fact, many scientists were fairly sure that there would be a red alert before it actually became very clear. So what are some of the main things, many things discovered at the time of the mapping of the human genome, but some of the most important and surprising things that were found were that humans have merely 20,000 genes, or maybe a few thousand more. We had thought we would have many more than that. Much of our DNA is shared in common with living organisms of all kinds. Much of our DNA, big surprise. Closely related species may have different genome sizes, and many plants have genomes which are much longer than that of the human genome. And what of most interest here today, that very large segments of the human genome are comprised of what was originally termed junk and shortly thereafter renamed as dark matter. In other words, whole segments, most of the segments of the human genome were at that time segments that we really didn't expect to find and didn't understand what was going on. Again, in the same year as Francis Collins' article appeared, this much-cited article appeared in Scientific American, and this article has been cited many, many times. New evidence contradicts conventional notions that genes are the sole mainspring of heredity and the complete blueprint for all life. Much as dark matter influences the fate of galaxies, dark parts of the genome exert control over the development and the distinctive traits of all organisms, from bacteria to humans. Some scientists now suspect that much of what makes one person and one species different from the next are variations in the gems hidden within our junk DNA. Environmental stimuli cause chemical tags to be attached to DNA, and we've heard quite a bit about this, and I will put it in my language now. These epigenetic activities control the expression of genes. This has been known for a good long time, but after the mapping of the genome, it became very clear that this had to be taken very, very seriously, not simply in terms of individual human development, but also in terms of changes over the course of the life course related to incidents of disease. Thus far, methylation is the best researched epigenetic tag, and it's been shown to play a pivotal role in delimiting activity by rendering chromatin structure inaccessible to transcription. These findings, and very many others, make it clear that hereditable information is by no means always already encoded in the DNA. Furthermore, genomes are best understood as dynamic reactive systems, responding to environmental stimuli originating inside the cell, outside the cell, and/or externally to the body, over the entire life cycle. With the advent of molecular biology and DNA sequencing, it became possible to detect important differences among the 10 million or so SNPs, single nucleotide polymorphisms, in the human genome. And recently, genome scanning array technologies have revealed numerous sub-microscopic structural variants among whole genomes that together contribute to variation within and across populations, greater than that brought about by SNPs. My emphasis is here is it is hard to grasp the enormous amount of variation that is present that has not been thought about to a great extent and can no longer be ignored with these findings. So, we've learnt that, or we are learning, that human variation is more subtle than had heretofore been assumed, and a great deal more remains beyond our understanding, a great, great deal. Most of this variation is inconsequential, important to recognize. However, a considerable amount has significance for human health, disease susceptibility, and for precision medicine. Signals that affect the activity of DNA, producing both similarities and difference among genomes, arise largely in the intracellular environment of the organism. Many of these signals are in turn responses to input from environments external to the cell and to the organism. An entanglement, as philosophers of biology and many social scientists and some basic scientists now are willing to argue, an entanglement of gene-environment interaction takes place ceaselessly from the time of conception. It's not genes and environment, or genes or environment. These things are tied up together in a very, very tight manner, which we are only beginning to comprehend. And they're concepts, of course, the whole question of what exactly is the environment or environments. Equally important, the genome of the conceptus on which environmental effects impinge is always already a product of the enduring effects of evolutionary, historical, cultural, and stochastic variables, stochastic being unpredictable variables, to which the DNA of previous generations have been exposed over eons of time. In each individual, then, genomic difference and similarities arise from two sources: DNA contributed by each parent to form the genome of the embryo, and this DNA itself has a history, of course, that it brings with it, and the effects on that genome made by environmental signals from the moment of conception. The situation is almost beyond bounds of being able to conceptualize it comfortably, the number of variables that one is thinking about or trying to think about. Obviously, some control has to be brought to this huge amount of variation, but it is also important to keep reminding ourselves of the complexity with which we're confronted. Obviously, genomes among families are closely shared, but equally important, genotypes present in larger social groupings and communities in which people live and conduct their lives result in a clustering of phenotypic features. Although, of course, today, massive global migration means that this situation is changing much faster than it did ever before. As Stephen Jay Gould, recently deceased, put it long ago, well before the post-genomic era, he said, "We cannot factor a complex social situation into so much biology on one side and so much culture on the other. We must seek to understand the emergent and irreducible properties arising from inextricable interpenetration of genes and environment." I coined the term local biologies in the early 1990s to highlight such clustered group variation. And I'm now going to give a couple of illustrative examples of local biologies, which today I now prefer to think of more as situated biologies. And if we ever find that we have a moment for questions tomorrow, the next day, I can talk about that. My hope is to stimulate some reflection on why it's important for precision medicine researchers to delve deeply into the reasons for and significance of biological variation in individuals and populations. My first example is that of the Apoe gene and the allelic changes it's undergone through time and space. The Apoe gene, all mammals have an Apoe gene, and it's essential for lipid transport and cholesterol metabolism. Today, the Apoe gene appears, with some exceptions, in one of three different allelic forms, Apoe 2, 3, and 4. But the percentage of these allelic forms varies across populations of people. And this example, with about 77 to 78% E3, is common to the so-called Caucasian populations, i.e. white populations. The distribution, as I'll make clear in a moment, is very different in other populations. This is really significant. We have to stop thinking of us all. We are all the same. We are all human. We all have incredible human attributes, but we do have significant difference, which has nothing to do with better or worse, good or bad, but we do have some very basic biological differences which derive from the environments in which us and our ancestors have lived for great lengths of time. So these three alleles differ from one another by only a few amino acids, i.e. a very small amount, but they have significant physiological consequences. So one really does have to delve deeper. One has to think about variation in genes very, very significantly. The presence of, in the case of the Apoe gene, the presence of one ApoE4 allele accelerates the age of onset of AD, that's Alzheimer's disease, by about 3 to 4 years. For carriers of two ApoE4 alleles, the age of onset may be 8 to 10 years earlier than non-carriers. In other words, the E4 puts one at risk for Alzheimer's, but only at risk. It does not determine anything. Research has shown that approximately 50% of individuals who are either homozygous or heterozygous for E4 are never diagnosed with AD. Very important to keep that in mind. And the ApoE2 allele appears to be protective, but we're very short on research in connection with the E2 allele. Now, I want to go on talking about the Apoe gene for a moment. The Apoe gene is well conserved. What this means is it's existed for millions of years and is widely present in mammals, including all non-human primates. However, in mammals other than humans, Apoe exists in one variant only that resembles fairly closely the human E4 allele, but is not the same, not exactly the same. It was proposed in 1988 that the E4 allele is the ancestral mammalian form of Apoe, including that found in Homo sapiens. And the association of the mammalian E4 with the characteristic diagnostic features of Alzheimer's, that is, deposition of amyloid plaques and neuron atrophy and other neurodegenerative changes in the brains of apes and certain other mammals in the latter part of their lives, has been known for a good long time. The well-known primatologist, gone on too far. The well-known primatologist Robert Sapolsky co-authored an article in 1999 with Caleb Finch, whose specialty is the neurobiology of aging, in which they postulated a connection in humans between the so-called grandmothering hypothesis and selection for sporadic mutations of the ApoE3 allele during the early history of Homo sapiens. The grandmothering hypothesis postulates that as a result of the extremely long dependence of human infants after birth, it became adaptive for grandmothers during their post-reproductive life to contribute extensively to childcare, leaving younger women free to forage for food. Similarly, the existence of menopause, virtually limited to human females, not quite, but almost virtually limited, suggests that a post-reproductive life in women is biologically adaptive. Finch and Sapolsky claim that if the grandmother role is important, and there's definitely evidence from hunting and gathering societies that this is the case, then a predisposition for dementia and cardiac disease conferred by the E4 allele would be selected against. This may in part account for why the E3 allele is today by far the most common among human populations, particularly among the so-called Caucasian white population. It's equally possible that genetic drift, of course, accounts for an increase in the E3 allele, and not everyone agrees with Finch and Sapolsky. But on the other hand, these researchers have also shown that in Africa, the E4 allele confers resistance to infectious disease, notably hepatitis C. And further, it's been shown that among younger people, E4 assists with memory retention. So we've got some very complicated things going on here. This kind of thing is not unknown in the world of genetics. And it's well known that the Apoe gene exhibits what's called Pleiotropy. That is, it can have two or more apparently unrelated effects. And findings to date strongly suggest that Apoe is an example of antagonistic pleiotropy, in which a given allele enhances the probability of maturation and reproduction, but in later life, it can predispose carriers to physiological and/or anatomical degeneration. And it looks as though this is what we may have with the E4 allele. The Italian geneticists, Rosa Maria Corbo and Renato Sacchi, carried out extensive research in the late 1990s to examine the worldwide distribution of the Apoe gene. And they found that among Pygmies, the Khoisan, indigenous peoples of Malaysia and Australia, Papuans, and some indigenous North American peoples, and the Lapps, that the E4 allele was and still is the most prevalent. Corbo and Sacchi noted that these are groups of people whose subsistence economy was until relatively recently predominantly that of hunting and gathering, and that they have lived in situations where food shortages are common. They go on to postulate that the E4 can best be understood as what is known as a thrifty gene, because it assists with a higher absorption of cholesterol that would be protective in times of food scarcity. These researchers suggest that the E4 allele may have become disadvantageous only after the adoption of the sedentary lifestyle that so many of us now essentially live. And a change to diets rich in carbohydrates and fat and low in fiber could also be a major problem. Their conclusion was that in a westernized environment, it becomes disadvantageous to carry E4. The question of which Apoe allele came first remains unsettled, but these ongoing arguments make it clear that susceptibility genes, such as Apoe, have what I would call a deep history, encompassing two time dimensions, evolutionary and historical, and their expression in any one individual or group of people depends upon local environments. And we would do well to keep this in mind. This is not yet the whole story. It's unusual for genes to exhibit only 3 alleles, and other Apoe alleles do indeed exist, several of which are in the regulatory area of the Apoe gene, but they are rare. And furthermore, molecular biologists have carried out comparative research in which they looked for and found substantial heterogeneity within the E2, E3, and E4 alleles, depending in large part on the population in question and its geographical location. This could account for a great many of the contradictory results that have long plagued population genetic research in connection with the Apoe gene, and that caused difficulties, really huge difficulties, in clinical trials. Cross-cultural research has brought about some interesting insights. What is known as the Indianapolis Ibadan Dementia Project, headed up by the psychiatrist Hugh Hendry, it's been running for over 17 years, and he's been working together with Nigerian collaborators. And they have come up with some very interesting results. The incidence of Alzheimer's and dementia among Yoruba is less than half that found among African Americans, even when controlled for age. Samples of over 1,800 African Americans and 2,200 Yoruba showed that the frequency of the E4 allele is not significantly different in these two cohorts. Furthermore, Yoruba have a lower incidence of both vascular disease and vascular risk factors, including hypertension, than do African Americans. And significantly, cholesterol levels and lipid levels are much lower among Yoruba. To date, this research has concluded that the ApoE4 has a significant but weaker effect on incident AD and cognitive decline in the Yoruba than in African Americans. And the data appear sufficiently robust to conclude that risk-reducing factors in Africa and risk-enhancing factors in North America must be implicated, among them other genes, many of which are very low explanatory power individually, diet, environment, and other unknown, completely unknown factors. So clearly, in-depth population research on gene functioning in context is crucial, and both whole genome sequencing and close attention to environmental variables past and present is needed. Furthermore, pleiotropy ensures that age must always be taken into account. Genes are shapeshifters, and even the best research of genes, such as ApoE, is far from well understood. And I want to take up a second example very briefly in closing, because this also is quite a shocking example in some ways. Globally, more than 2 million children die from malnutrition every year, and recent research has revealed remarkable findings about biological differences between infants who suffer from what is known as marasmus, which is a wasting disease caused by profound generalized malnutrition, as opposed to kwashiorkor, which is malnutrition caused by severe protein energy malnutrition, causing edema and anorexia, among other severe symptoms. This impressive study was carried out in Jamaica. It commenced in 1962 and continued for 30 years. And this is the kind of length of research data that we need. During this time, over 1,100 infants with severe acute malnutrition were admitted to the University Hospital, Kingston, and it was found that these infants diagnosed with kwashiorkor had considerably higher birth weights than did infants diagnosed with marasmus. The authors concluded that mechanisms associated with physiological plasticity, as they called it, are operative in utero, and that these children have distinctly different genetically influenced types of metabolism. Wait a minute, we'll go back. Of the two conditions, children more often die from kwashiorkor, and children diagnosed with marasmus endure much greater wasting of their flesh, although their survival rates are much better than those with kwashiorkor. Researchers characterize marasmus as metabolically thrifty and kwashiorkor as metabolically profligate. They propose that in the case of children with marasmus, when the maternal diet is low in nutrition, fetal metabolism in utero in effect anticipates a postnatal environment of scarcity, and low birth weights are assumed to be evidence of this process designed for survival. So based on a hypothesis known as the mismatch pathway, which was where are we here? Oh, I seem to be going. Okay. Based on a hypothesis known as the mismatch pathway, it's posited that evolved adaptive responses of a developing organism to anticipate future adverse environments can have maladaptive consequences if the environment is not what has been biologically anticipated. In other words, if fetuses and young infants are exposed to nutritionally deprived diets, their bodies may be epigenetically prepared to deal with deprivation as they mature, a situation that can cause havoc in energy-rich environments. Added to which, of course, maternal diabetes, maternal obesity, and infant overfeeding increase risk of obesity in adult life. It may well be that in the current era of the Anthropocene, with its inordinate rate of global change resultant from human activities, both environmental and social much of it involving extraordinary violence and dislocation of one kind and another, that mismatches will increasingly be taking place between environments to which human populations are reasonably well adapted biologically and the actual lived environments in which millions of people are forced to live. The very function of genes changes depending upon environments, but we humans are moving or being forced to move at such a rapid rate as has never been the case before, and it looks as though we are bringing about serious biological dysfunction on ourselves. I'm going to finish with one final point. The Human Microbiome Project is proving to be extraordinarily rich, leading Tom Insel, the director of the National Institutes of Health, to proclaim recently that at least from the standpoint of DNA, we are more microbial than human. This has enormous implications for our sense of self, he added. 2 million or more genes of commensal bacteria, viruses, and plasmids can make our 20,000 or so genes seem very paltry. This complex ecosystem is meticulously tuned to adapt continuously to the vagaries of human life and adds yet another challenging dimension to our growing knowledge about the reactive genome. Obviously, precision medicine will have to take microbiomes into account as key actors, as key contributors to human variation and to individual health and illness. And I will leave you with this slide, a very provocative slide for which you might want to think about a future conference. Thank you very much.